Hello, and welcome to this episode of Genomics Lite. My name is Jack.
And I'm Fran, and we both work in the science engagement team at the Wellcome Sanger Institute. Our roles to enable everyone in the world to connect with and learn more about genomics.
If you haven't joined us before, Genomics Lite is a series of short sessions that share the science that happens on our campus, which is all about DNA. This is the first video of our autumn series, exploring some key questions about genomics that we get from people like you.
So the series is streaming live on YouTube. So welcome if you're joining us on the live stream. And all of our Genomics Lite videos are available on our YouTube channel.
This episode is all about cells, what they are, how they grow, and what happens when that growth goes wrong. We'll be joined by two fantastic guests who both spend their time working with cells in the laboratory, studying how that growth process happens. Now, a quick trigger warning that our guests use cells to explore the science of birth and the science of cancer, and we'll be talking about both those topics in this video. But before we get started, Fran, what do viewers need to know? Can you first define for us what a cell is?
Well, a cell is basically the smallest unit of life. So kind of like a building block that makes up all the living things. So some organisms like bacteria are made of just one cell, while other larger organisms like us have trillions working together. So you could look at it as each cell has its own little factory setup inside. So lots of different parts doing jobs to keep the organism alive and running smoothly. If a human has 37 trillion cells. And what's interesting is that there's hundreds of different types of cells, all specialized to do different jobs. So if we were to zoom in on these cells, like using a microscope, they all look quite different. So if we were to zoom in on skin cells, they look a little bit like a brick wall, creating a barrier to protect our bodies. Now, if we were then to look at, say, muscle cells, they're quite long and stringy, and these contract and relax to help move our muscles and our limbs. And then if we think about our brain cells, which are called neurons, they look a little bit like trees with branches for sending lightning fast messages.
Okay, so you've given us a picture of the cells there, and you've said that human beings are made of 37 trillion cells, huge number. So how did we get there? We didn't begin as 37 trillion. So what was the journey we went on?
So yes, we did. Yeah, we all started with one. So it's like, how do we turn into trillions? So it's worth also pulling apart the word grow in our title. So cells don't just keep getting bigger and bigger, because that would actually be really impractical. Instead, they divide once they reach a certain size. And this process is called mitosis. And it's where that one cell splits into two identical ones, and then they divide again and again and again. So with this process repeating, they multiply and they start to specialize into cells that we need to, let's say, form a complete organism. So that's how you go from one tiny cell to trillions. But of course, each of those steps is quite complicated than that. And so we'll explore that a little bit more with our guests today.
Right. Okay. What about how do they know when to grow and how to do that growth, that mitosis?
So there are a lot of chemical signals and checkpoints that act, I suppose the best way to say it is they act a bit like traffic lights, and these can signal like cell, okay, it's time to grow or divide, or nope, stop, not yet. It all depends on what the body needs. And there are actually even some signals that tell the cell to die. So everything we talk about on Genomics Lite connects to DNA, and it is in that DNA that a cell finds all the instructions it needs about when to grow or when to divide and how to essentially behave once that division has happened. So every step from growing to dividing is super, super organized and carefully timed. So it's a bit like a perfectly choreographed dance.
Okay, so we've got the image of this perfectly choreographed dance in our minds under the microscope, and I think this is the right time for us to introduce our first guest. They are a rather special collection of a few trillion cells, and their name is Alisha Dordi. So Alisha works in a team at the Wellcome Sanger Institute called Cellular Services, and her job is to grow cells in the laboratory that are then used for scientific research. So what a perfect person to help us answer this question of how cells grow. Hello, Alisha.
Hello, thank you so much for having me.
Oh, well, now, Alisha, we've heard from Fran what a cell is. these building blocks, and what's meant by growth, this mitosis, this cell division. But you grow cells for a living, so I want to hear about that process. What does a cell need in order to be able to grow?
Yeah, of course. It's a brilliant question. So cells, whether they're in the body or whether they're in a lab being grown artificially, they all need a key few things to grow. And you can imagine that's the building blocks of life. They need the right nutrients, they need the right temperature, they need the right signals from their surroundings that they're growing in. So in the body, those would come typically from your bloodstream. But in the lab, of course, we're not able to provide that. So we have to do that artificially. So what that is basically growing cells in a nutrient-rich environment, which we call growth medium. And that's typically, it looks like a soup full of amino acids, a soup full of proteins and growth factors that act like food for the cells, essentially. And it's very important that we also keep them at body temperature because at the end of the day, we're trying to mimic the human body as close as possible. So we also control things like gas exchange. So for example, the levels of oxygen and carbon dioxide that they come into contact with. So all of these things put together are really how we can control how they grow in the lab and kind of make them feel like they're at home. What we're doing is we're recreating as closely as we can that they would actually experience in the human body.
It sounds like quite a beautiful setup. You're trying to create this lovely, warm, nourishing environment for them. I want to step us through this process though now. So can you describe for us, first off, what is your starting material? What do you begin with to grow these cells?
Yeah, of course. So cells that I work with specifically, so I work with stem cells, and they are the, forming cells that progress to then specialize to become different types of cells. So they are in many ways the building blocks of, how different cells, for example, turn into neuronal cells, or some become more specialized, like the cells I work with become placental cells, so to develop into pregnancy. So it's quite fascinating because the cells I work with are belonging to the placenta. So the placenta is like a temporary organ, and it grows quite rapidly as well to establish a connection between the growing baby and the mother. So, and then that placenta disappears after birth. So in essence, it is the first formation of life. And then what's also remarkable about them is that they can invade into maternal tissue. So they have like specific qualities that allow them to do that. And we call that as differentiating. So what differentiating means is if you sort of think of it like a classroom full of students, each student, you have identical students in a classroom. One might become a doctor later on in life, one might become an engineer, another might become a teacher. So you can kind of apply the same concepts to cells in that they differentiate, hence they specialize in different areas. So the cells I work with do that essentially. However, we're really trying to understand what makes those cells so special, what gives them the ability to invade it during pregnancy, and how does that, form a baby. That's what I study in the lab.
I think that's another beautiful image that we have here of these stem cells as primary school children or something like that, just setting off on their journey to specialize. I've got a lovely vision of that. Where are you aiming to get to with that growth? Because you're not taking these through to grow a whole animal, are you? Where do you get to?
So I work in the organoid field. So what an organoid is it's basically a miniature version of organs. To clarify, it's not that we're actually growing a whole organ in the lab because that I won't say that's impossible, but we're very far off from being there. What organoids are is basically they're 3D miniature versions of organs. So they contain all the same cell types that you would have in an actual organ, except we grow these in the lab. So our aim is to generate 3D structures of what happens in placental development. So the way these cells communicate, how they arrange themselves, how they actually carry out the invasion into the uterus during pregnancy is something that my team does. And all of that basically comes back to the trophoblast stem cells that I work with. So we actually generate the organoids from these stem cells. Now that process of differentiating them comes with, giving them the right things to grow. So nutrients that they need to form these 3D structures, and to mimic what happens in early pregnancy as well. So organoids kind of give us a window into what's going on in the very first few weeks of pregnancy. And that can't really be easily studied in people because it happens so deep inside the human body. And it's quite difficult to replicate that outside in a lab. So we try to use organoids as our model to do that.
A simple question to help me understand where you're aiming for with these organoids is how do they compare in size to a placenta that you would see in a living human?
So the organoids are quite small. So they're bigger than what a single cell would look like, because they're a collection of thousands of cells put together to form a 3D structure. You can see them by eye sometimes, so they can range in size. The ones I work with are typically a few millimeters across in size. So they're significantly smaller than actual human tissue, but they do contain the same cells that you would find in the actual human body.
I've got some images that I can share with people here. And I'm just going to put these up now. Perhaps you could talk us through what it is that we're seeing in the window here. So if I just make this full screen, sorry, I've got your, let me just put this as full screen. We're going to put these up, and I'd just like you to help us to understand what it is that you'd be seeing if you were looking at this under the microscope.
Yep. So this is essentially my starting material. So what you're seeing in the picture here is a 2D trophoblast stem cells. So what trophoblast stem cells are is that in early development of a fetus, these cells would go on to form the placenta. So I'd like to make an important distinction here in that trophoblast stem cells are different to embryonic stem cells. So embryonic stem cells are what the cells that you would expect to go on and form a baby in the later stages of pregnancy. So these cells aren't that. These are the cells that form the placenta, which is the bridge between mother and baby. So that's what you're looking at here. These 2D trophoblast stem cells, which I start with, I culture them, and I grow them up to then form organoids.
And we can get, for viewers, you can see that scale bar down at the bottom there. If you can't quite make out the text, that's 400 micrometers. So a little less than half a millimeter is the length of that red line. So these are really small cells.
Very small, yes, micrometers, yes.
Yeah, if I go on to the next image, we should be able to see. What are we seeing on this one, Alisha?
Yes, so this is what happens when you allow those trophoblast stem cells in the slide previous to grow a bit more and self-organize into organoid structures. So these are actually 3D structures that sit in a sort of jelly-like substance, which we call matrigel. But essentially, it's a gel that allows them, it promotes the formation of these 3D organoids. So what you're looking at here are what we call placental organoids, and they mimic what you would see in early stages of pregnancy.
And I know you've used that distinction 2D and 3D a couple of times now. I guess what lots of people will have seen on television or perhaps at school will have been growing maybe bacterial cultures in a flat membrane in a petri dish or something like that, but these organoids are crucially 3D. They're not on that flat surface. So just help us understand what do they sit in the lab? What would we see if we were looking at with our naked eye?
So you would see, so all of our the labware that we use in the in the lab is plastic. So these organoids actually just grow on plastic. The key difference between the 2D and the 3D is that the 3D cells are sort of submerged in a jelly substance. So what you would see is you'd see that jelly substance, which is like a 3D blob on the surface of that plastic. And these organoids actually are embedded within that and grow within that jelly. So you can see them by naked eye, but that's typically when they're a bit bigger, like how they are in this picture. Typically, when you first do an experiment and you get them to grow into the organoids, you don't see them very quickly. It takes a few days for them to form. So the ones that I work with, the transition period is typically a week before they start looking like this.
Mm. And that is relevant, I suppose, for our question, how do cells grow? Because cells don't tend to grow, at least not in humans, in 2 dimensions, do they? They do grow in these 3D tissues in our bodies. What does that mean in terms of what it allows you to do when you study them? Why is the 3D important there?
Because it's a more representative model of the actual organ that we're trying to model. So as you know, we 2D cultures have been around for years, and they're very well researched, documented in science. However, the thing that 2D cultures lack is that 3D interaction between cells. Now, we've been able to research organoids for a couple years now, and it still is an emerging field. So they're a much better, model for us to use and actually predict how cells are going to respond to even drugs, different types of treatment, investigate gene therapy, and they're just they're a more representative model of what happens in the human body. And the other benefit I would say is that we are trying to diverge away from using animal models. So because organoids are a relatively easy model now for us to use, and it doesn't rely on you testing animals, they are the more preferable technique, in my opinion, to go forward with.
Mm. I know that lots of our viewers will feel strongly about animal testing, and so it's useful to hear that this method means that scientists don't need to do that as often. I've got one more picture to show, and I'd just like you to explain what we're seeing in this stunning microscope picture, really. So what do we have here, Alisha?
Yeah, so as you can see, there's a drastic difference between the previous image and this one. What we do to make the organoids look like this is that we differentiate them. So when you actually have a pregnancy in the human body, there is a fertilized egg that needs to fuse to the mother's uterus. Now that fusion is the building block of the placenta. Sometimes you don't get that fusion that happens very healthily, in which case it can lead to downstream pregnancy-related diseases such as sudden miscarriages, ectopic pregnancies, placental fetal growth restriction. So you can get syndromes like that occur. So that invasion process is very important, and it's the most important thing that will happen in those earliest phases of pregnancy. That's what we're trying to recreate in the lab. So we're trying to build those outgrowths. So what you'll see in this image is that the main striking difference is that they have these projections. So it kind of looks like a little bit like a fried egg almost. So they develop these outgrowths, just projections from the center of the organoids. And this is what happens in the actual during actual human pregnancy. They try to attach to the uterus, and that's what we're trying to investigate. How does that happen? And if it goes wrong, what's going wrong over there?
Great, thank you. I'm going to stop screen sharing now. But what we were seeing there, I suppose, was that real cell growth and how cells grow in action. We're seeing a really important stage there replicated in your organoid structures. I think I'd like to ask a little about the timeline that you go on with these cells. So how long does it take for you to get from those tiny individual stem cells that we began with to this point here where we saw the organoids starting to differentiate?
From start to finish, it takes around a month. So not a significant amount of time, actually, because we do start from a frozen bank of these stem cells, and then they have to grow, which takes about 2 weeks for them to grow. And then to form the organoids, that also takes another 2 weeks. So on average, it takes about a month to get to the stage that we just looked at. We also differentiate them, which means we try to form those outgrowths. So that takes some additional time because we have to supply the cells with different types of media that they grow in to promote that differentiation. So on average, about a month.
Brilliant. Thank you so much, Alisha. That was so interesting. And those images were just fantastic. So I think this is the perfect moment to introduce our second guest. So Yousra Belattar works in the Gale lab at the Wellcome Sanger Institute. And Yousra also grows cells, but this time she's studying cells that aren't growing quite how they should. And these are cells from cancer tumors. So hello, Yousra.
Hello, thank you. Thank you for having me on the podcast today.
So we're going to get on to cancer shortly, but in order to talk about that, we probably need to go sort of inside the cell and think about what needs to happen inside the cell to make cells grow, essentially.
Well, essentially, Fran, you explained it perfectly in the beginning where you you had the traffic light system, and you did touch on many things that I was going to talk about, but we'll hopefully delve in deeper. So like you mentioned, inside every cell, there is a nucleus, and in the nucleus, it has the DNA, which has the manual, the instructions for how the cells need to work, how they need to divide and grow. And what happens is when these cells receive a signal. So as Alisha mentioned, cells need to grow and have certain external environment to help them grow. So when they receive these signals, for example, from nutrients, growth factors or hormones, it initiates a specific signaling pathway. I'll give you an example of one is the MAPK pathway. So if you can imagine a single molecule called a growth factor, and it's just floating around the cell, and then it recognizes a receptor, or like you could say it's like a little home or its working base, let's say, and it will bind to that receptor. And then what happens, it's almost like a domino effect. So it's like a chain of reactions where you'll have the growth factor will initiate and send messages through the cell to activate certain proteins, for example, RAS or RAF, and these are all different proteins. And when it reaches to the end protein, it's the final signal that reaches the nucleus where the genes are switched on, and it's like, okay, let's get going, let's start working, let's start producing new proteins. And the proteins, you can think of the kind of like the cell workers, so they actually do the work, and then they initiate the cell division. And as many of you might know, the simple 4 stages of cell division. So you have the cells grow, start making new proteins, and then the DNA is copied where the new cells receive the exact same DNA material, so the DNA code, and then these produce 2 daughter cells. And within that process, of course, if you think about cells, they're very complex and everything needs to kind of work in unison. There's a lot of checkpoints. So you think of like there's police. So there's like P53 CDKs. So kind of like police, just to make sure that everything's working efficiently and working correctly. And what happens is if things don't work correctly, then they can do certain things to kind of manage that.
Brilliant. So you said that obviously they're not doing things properly or things don't working. So your role is to sort of look at cells that aren't growing in perhaps the way that they should. So why is that? How does that come about? Why do the cells not follow the normal rules, so to speak?
Well, if you think about it, these cells kind of rely on very precise controlled systems, and they are controlled by genes. But what can happen is that in that process, so if you think you're texting somebody and you want to write something, how many typos do we have? So many typos. So in that process of these, this control system of these gene functioning systems, there can be things that can go wrong. So mutations can occur. So changes in this genetic code in the DNA, and this can then cause these control signaling pathways that I mentioned before to be disrupted, or to keep growing uncontrollably. And it can lead to many kind of effects downstream, so whether it's cancer or disease.
So with particularly, as you mentioned, cancer, so how cancer is very complicated, but how can, again, these mutations lead to cancer developing? Is it one change, or does there have to be lots of changes?
So kind of it happens when, like I mentioned, there's a mutation, so there can be like a single change in the DNA sequence, so from an A to a T or a C to a G. And this can happen where when cells normally divide, so I mentioned these checkpoints, there's also checkpoints to make sure that the DNA is happening, the DNA division is happening correctly. So there's also DNA repair mechanisms. So if something's going wrong, there can be a signal that says, okay, stop this process or fix this process, or can lead the cells to just die. And you might kind of mention that apoptosis, so the cell just initiates like self-destruction, essentially. So what can happen in cancer is all these mechanisms can stop working. And if you think about a car, you're driving and you have your foot on the pedal, and if there's no brake mechanism, then that's dangerous, and things can go wrong, and horribly wrong. So when there's no control system, that kind of causes cancer. So the mutations that occur that kind of lead to apoptosis, so self-destruction not working, so meaning that kind of bad cells just keep growing, they don't self-destruct, or it can cause these cells to then keep growing and then invade other areas in the body where they're not meant to be, and that can cause a lot of disturbance. And these mutations essentially can happen for many, many reasons. Scientists are still working to this day to find out new reasons of why mutations can happen. So it can be simple mistakes, random mistakes. Like I mentioned, use the example with the phone and texting. When the cell divides, it literally just copies the DNA. And sometimes this copying process, errors occur. Also, there's environmental factors, which is very, very important. So, for example, smoking, or kind of intoxication, or certain chemicals that you're exposed to, or prolonged exposure to kind of UV light from the sun, like radiation, and even certain infections or bacteria can actually cause cancer and changes in the DNA.
Gosh, it's just so complicated, isn't it? So what particular kinds of cells do you work with? I mean, are there particular cancers that you focus on?
Yes, so I work mainly on blood cancers and lung cancers, and also colorectal cancers, and they're all very different. They all stem from patients who have had these cancers. So, for example, chronic myeloid leukemia is one of the certain blood cancer that I've worked with, and also lung cancer, and lung cancer is a very prevalent cancer here in the UK.
So with the leukemia, cancer is quite different. So a leukemia would be quite different to a colorectal cancer. Do the cells look different? And things like that?
Yes, so kind of Alisha mentioned kind of how cells look. So I work with 2D cells. So if you think about, if it's a blood cancer, how do blood cells exist in the body? They exist, they're in suspension. So we call them suspensional cell lines, because they exist, they're just floating around. And in the microscope, when you're looking down, they're floating around. But for example, lung cancer, if you look at the lung, it's in a fixed place. So they're called adherent cell lines. So what happens in the flask, they would stick to the flask at the bottom. So the way they're dealt with is very, very different. But I know organoids are even more complex when you work with, but yeah, they are quite different, and they need different nutrients, different media, and yes, and it also depends what you're doing with them as well.
Brilliant. So cancer research, it's been going on for many, many years, and the way that we study cancer has really changed quite a lot and quite rapidly because of all the changes in technology. But what is different about the ways that you and your colleagues are studying cancer? Are you using some cutting edge techniques to do this?
Yes, absolutely. So our team is using CRISPR technology, which within the last 10 years has kind of ramped up significantly and really led to a lot of novel findings. And we use tools a tool called base editing and prime editing, where if you think about it, like you mentioned, so we use molecular scissors, where it helps us change individual or a group of letters in the DNA sequence. And essentially, we kind of do this so it helps us kind of mimic what we're seeing in patients. And we do this ultimately to find kind of reasons for drug resistance. So for example, in chronic myeloid leukemia, so what type of blood cancer, we have found that there are the first drug that is prescribed to patients, more than 30% of them are completely resistant, so they don't work. So we're trying to mimic all this, the variety in these patients, using these tools so that we can test to see which drugs work and which drugs don't before they even get to the patients.
Yeah, that's fantastic.
I'd love to explore that a little more, Yousra, to connect it to this sense of cell growth. So you drew this picture for us of these ways that the cell is controlling whether it grows or whether it doesn't when it's a healthy cell, and that you're using CRISPR then to perturb that, to kind of prod it and find out what elements within a cell that isn't growing properly are responding to drugs and aren't responding to drugs.
Yeah.
If you were looking at those cells that were resistant and weren't resistant under a microscope, would you be able to see any difference from how they look? Or do we need to use this process that you're using to unpick what's going on there?
Well, if you think about it, we're using, for example, one cell line that's derived from one patient. And that patient, so for example, if you have two people who both have this type of blood cancer and you test them with the same drug, you can find that one patient, the drug works, and the other patient, the drug doesn't, even though they have the same cancer. But that's because we are all genetically different. We develop mutations over time. And actually, when you get certain cancers, the cancer is very smart. It finds ways to avoid being killed. So what it does is that, okay, there's this drug, I'm going to mutate the exact same point where this drug binds, so that you have to then discover another drug where it doesn't mutate to that same point. So different patients have different mutations. And what we want to do is essentially create as many mutations as possible in these cell lines, test these drugs, and see whether, okay, this patient A with this kind of genetic code, maybe this drug will work, maybe this wouldn't, rather than us doing it with patients and then kind of working retrospectively.
So you're trying to get ahead of the cancer.
Basically,
you're trying to figure
Yeah.
out the solutions
Trying to
before
be
the
10
cancer
steps
gets there.
ahead. Absolutely.
Yeah. Okay. So this feels like one really good example of how studying the way that cells grows is giving something really useful. It's helping us develop these drugs that are gonna.
Yeah, absolutely. And sometimes we find that there's mutations that haven't been found in patients. So actually, you can work ahead and you can see, okay, if this patient has this mutation, this drug won't work, or this drug will. So it'd be resistant to this drug, or it'd be sensitive to this drug. And under the microscope, you can see when you introduce these mutations, when you add a certain drug, you can see the cells die. And I'm sure, Alisha, you know what a dying cell looks like or a dead cell looks like. So, yeah.
You're making this picture a tragic scene here, but these are cancer cells, so we don't want them around.
No, absolutely.
But this is, yeah, I'd like to bring you back in at this moment, Alisha, because you're almost looking at the opposite end here, aren't you? You're at the very beginning of the journey of cell growth. When the placenta is first forming to support that young fetus. How does studying the way that cells grow at that point, why are you looking at that area?
Yeah, of course. So it's actually really important that we understand what the good looks like first, because the area I'm working in, it's a little bit misunderstood what happens in a healthy pregnancy. See, pregnancy is well documented and we know what it looks like, but on a molecular level, what's going on there? There's so many things that are involved in how a fetus implants into uterus, what all the mechanisms that come together to allow that to happen is quite under-researched, and there needs to be more focus on it. So it's really important on one end to first understand what's going on there. The other end is that so we can compare it to actual patients, for example, by taking biopsies from ectopic pregnancies, for example. So comparing both of what we know is actually happening and what should happen to actual patient samples taken from donors, for example.
So you described earlier how studying these organoids allows you to observe and to understand aspects of cell growth that you simply couldn't do in a living person and a pregnant woman. But what you're saying is you still need to check back. You're not just looking at these cells that you're growing in the lab. You need to make sure that what you're growing is the same as what would happen. Is that
yeah, exactly. The drivers behind a successful implantation or a successful invasion of the growing fetus, that those molecular drivers and what signaling pathways are responsible, that's quite under-researched. So that's an area definitely of a lot of groups that want to research that area.
And that signaling pathway that you've mentioned there, that's the same kind of language that we heard Yousra talking about earlier in terms of the cells that she studies. Are there fundamental differences in terms of what these placental cells are doing to grow, or are they behaving in a different way than cells of a fully formed and birthed organism?
Yeah, so the stem cells actually differentiate so they can move into the maternal tissue. So it's quite, for example, with Yousra's work in cancer cells, they acquire different mutations to help them avoid the immune system or to hijack cell signaling pathways or areas where they would stop growth. Cancer is very smart, as Yousra said, and it avoids that. But the cells I look at, they actually want to move to other areas of the body. They want to invade into the maternal tissue. They want to, establish that blood flow. So that's one of the key differences that is really amazing that these stem cells are able to do. They establish that network between the mother and the growing baby.
Oh, fantastic. It's just so interesting. And it feels quite futuristic, some of the technologies that you're using. Obviously, you're using organoids, Yousra's using CRISPR. So where is the science going? It obviously seems this is all developed quite quickly, but what's going to happen in the next few decades, Alisha, that's going to really transform your field, do you think?
I think it's the long-term vision is definitely to help understand why certain pregnancies and why certain mothers suffer with some complications. The end goal would to be, I think, identify those early warning signs of a risky pregnancy, understanding the root causes behind that, and then one day maybe even be able to prevent some of the complications before they even happen. That would be the gold standard and the goal for sure. What's exciting with working with the placental stem cells in the lab, we can, it's a collaboration with with another group here based at Sanger. They're looking at identifying certain molecular pathways and genes and signaling pathways that are responsible for the healthy pregnancies versus unhealthy pregnancies. So it's working with live actual patient samples as well. So it's not just limited to organoids. This can be applied to so many different types of samples. And I think in the future, hopefully, work to correct some of those complications that happen. Fingers crossed.
Fingers crossed indeed. Now, Yousra, do you think there's any new exciting technologies around the corner that's going to help with your research, that's going to take it forward to the next level?
Yeah, of course. I think similar to the end goal is to improve patient outcome, and actually, yeah, prevention is better than cure, right? So if you can prevent this is amazing. And it has happened with CRISPR, so using CRISPR to actually treat, for example, sickle cell disease, which is a genetic disease, and it's worked, and it's provided a lot of hope for research. And as well, we are in the era of AI, so we need to think about how we can use AI to help us even in science. So, for example, our aim is to hopefully use AI to build kind of machine learning models that can help us actually predict. So instead of waiting for the patients to be treated with this medication, actually, how can AI help in this process? So yeah, and AI is proven to be a very, very powerful tool.
AI not something to be feared, but something to really sort of help us to go forward with our scientific research.
And as scientists, we use it a lot, and I think AI is probably the future, and yeah, we should use it to help better science.
I know we're going to be returning to AI in some of our future episodes. It's definitely a theme, I think, across this science as it continues to grow and to scale. Well, I'm afraid that's all we've got time for on this stream today. It's been a fantastic journey we've gone on, right from single stem cells on a plate through organoids and CRISPR, and to a future full of possibilities. So thank you very much, Alisha and Yousra, for joining us today.
Thank you.
And thank you so much for watching. We hope you've enjoyed this session. If you did, please subscribe to the YouTube channel, Your Genome, so you don't miss an episode. And we've got lots of other content for you to explore on that channel as well. Genomics Lite will be back next month. We're sticking with the theme of cells, and we're going to be asking, how many cells do I have? We'll also be sort of looking at those numbers that we mentioned at the start of the episode. We'll be delving into the different types of cell types there are in the human body, but also we're going to be finding out about an amazing project that is looking to map them all, and that's called the Human Cell Atlas. But until then, goodbye.